Hello, this is a video on the organelles of the cell. There's a lot to go over, so let's jump right in. Here is a basic cell that we will fill in over the course of the video. We have the plasma membrane of the cell, which we covered in the previous video. Remember that it's a phospholipid bilayer, but I'm just drawing it as a single line for simplicity's sake. Then we have the cytoplasm, which is a gel-like liquid that makes up the inside of the cell. You might see more words with the prefix cyto in the future, so keep in mind that just like cyte in keratinocyte, it's really just another word for cell. Next we have the skeleton of the cell, aka the cytoskeleton, which gives the cell its structure and shape. The cytoskeleton is made of three differently sized molecules that we call filaments, which are long chains of proteins that form a thread-like structure. In the order from smallest to largest type, we have the microfilaments, intermediate filaments, and macrofilaments. Microfilaments are also called actin filaments and are generally found at the periphery or the edge of the cell, right under the cell membrane. They have many different roles, one of which is to provide the structure for small protrusions from the cell called microvilli. The main role of microvilli is to increase surface area, which is important for absorbing nutrients in the gut, for example. Macrofilaments are also called microtubules, which are found throughout the cell and, like actin filaments, have many different roles. We'll focus on three in this video. One is that they form what we consider highways within the cell. When things need to move from one place to another in the cell, they are usually being transported on top of microtubules. Another important function is that they form the centrosome, which plays an important role in attaching to chromosomes during cell division so that the chromosomes can be separated into two different cells. Last but not least, they form little eyelash-like structures called the cilia on the membrane of the cell. Similar to how eyelashes keep particles out of our eyes, cilia help sweep away particles, among various other functions. Intermediate filaments are any filament that doesn't fall into either category and won't get much focus. The nucleus is very important because all of our genetic information is stored inside of it in the form of DNA. We will learn more details later, but when the nucleus wants to make some kind of protein, such as all these filaments around the cell, it will transcribe the DNA into messenger RNA, abbreviated as mRNA. The mRNA will then leave the nucleus to be translated into protein. This brings in the next player in our cell. The ribosome is a molecule that translates the mRNA into amino acid sequences that make up a protein. Interestingly, it is itself made of proteins. There are two types of ribosomes. One is what we call free ribosomes, which are ribosomes that are just floating around the cytoplasm translating mRNAs. These generally make up proteins that will stay in the cytoplasm, such as the filaments that make up the cytoskeleton, or proteins that will go back into the nucleus. However, proteins that need to go to the membrane or leave the cell entirely take a special pathway, which is where the other type of ribosome comes in. These are bound ribosomes. This creates what we call the rough endoplasmic reticulum, or rough ER, because that's a bit of a mouthful. The rough refers to how the surface is bumpier due to the bound ribosomes. Because of this, one of the main roles of the rough ER is protein synthesis. The part of the ER that is not embedded with ribosomes is called the smooth ER. In contrast to the rough ER, the role of the smooth ER is to make lipids. The smooth ER also helps with detoxification in liver cells. Proteins that are made at the rough ER get packaged into vesicles. These vesicles can then travel from the ER to the Golgi bodies. You can think of the Golgi as the post office of the cell. It will take the proteins, process them or give them modifications if necessary, and then package and ship them to different parts of the cell. Some of these vesicles will make their way from the Golgi to the membrane, pulled along by specialized proteins that walk along the microtubules. The next organelle is the lysosome. You can think of the lysosome as the trash can of the cell. It has specialized enzymes that break down proteins or even organelles. To remember their function, think lysis, which means breaking down. Another thing to remember about the lysosome is that it has a very low pH, so it's very acidic. Peroxisomes are another organelle in the cell. Their function is to perform oxidation reactions. They generally use hydrogen peroxide to oxidize other things, which helps break down hydrogen peroxide into water. Since too much hydrogen peroxide is toxic to the cell, peroxisomes are very important for making sure they don't do too much damage. For these organelles, think peroxide. Last but not least is the mitochondria, most well known as the powerhouse of the cell. Mitochondria are unique because they have their own DNA separate from the rest of the cell. The theory is that once upon a time, the mitochondria was a prokaryotic cell that was engulfed by another prokaryotic cell, and they ended up working together in a mutually beneficial relationship. Specifically, the mitochondria was able to help the cell produce lots of ATP, which is what the cell primarily uses for energy. As you can imagine, cells that need to use a lot of energy, such as our muscles, have a lot of mitochondria. Hydrogen peroxide can be produced as a byproduct during the process of making ATP, making the peroxisomes a very important partner. This is where we'll end the video since we have now covered the main components of the cell. In the next video, we will talk more about ATP production.